Hi everyone, welcome back to class. It's learning time, so let's dive into the business of the day. Our study focus for today will be the dark phase reaction of photosynthesis, which is also known as the Calvin-Benson cycle or the C3 pathway. As discussed earlier in our previous classes, where we discussed photosynthesis, which we say is synthesis of organic compound from carbon dioxide and water, so in the presence of light, sunlight. So we were able to note that in that, for us to be able to get our organic compound, which in this case is our glucose, we'll be needing some main things, which include the CO2, carbon dioxide, and also water. Also in the presence of chlorophyll and also sunlight, which is the light energy. Now, and we also make mention that the dark phase reaction happens in the absence of light. That is part of what makes it differ from the light phase reaction. But what also now join them together is that the product gotten from the light phase reaction is what will be used by this dark phase reaction to be able to achieve their goal. And what is the goal of this dark phase reaction is just to produce our organic compound, which is the glucose. So the products we now get from our light phase reaction, which includes the ATP and also the NADPH, is now what will be utilized by the dark phase reaction. And in this dark phase reaction, it is now CO2 that will now start our reaction. So let's dive into the pathway proper. The plant we uptake CO2 from the environment, and they are able to achieve that process by opening up their stromata. So when they open up their stromata, and the stromata is located on the leaf, so when they open up the stromata, they'll be able to get CO2. So upon entry of the CO2 into the plant, this particular pathway will occur in the stroma of the chloroplast. The CO2 that has entered through the stromata into the stroma of the chloroplast will now be fixed to give us organic compound, which is glucose. So that's just the summary of this particular pathway, but it involves different steps and different reactions before we'll be able to get this our organic compound, which is glucose. So let's take this step one at a time. We have four major steps in the Calvin-Benson cycle, which include: one, we have the carbon fixation step. The second step is the reduction step. The third step is the isomerization or dismutation step, while the last step is the phosphorylation step. In carbon fixation step, what will happen is that CO2 will now react with ribulose 1,5 bisphosphate, which in short form we call the RUBP. Because of limited space here, I will not be able to write it fully, but after the class, I will list out the steps and also the enzyme that catalyzes each reaction in the step. So let's continue. CO2, which is our carbon dioxide, will react with ribulose 1,5 bisphosphate. The enzyme that catalyzes this reaction is ribulose 1,5 bisphosphate carboxylase. The short name of that enzyme is refers to as Rubisco. So when this enzyme catalyzes this reaction, it will now give us two molecules of 3-phosphoglycerate. This process now occurs 3 times. That means, we started with one molecule of CO2. So that means after this process that we've gotten two molecule of PGA, then another CO2 we enter. So we get another two molecules of PGA, making 4 now. Another CO2 we enter, so we get another 2 molecules of PGA, making 6. So in total, for this carbon fixation or the carboxylation step, we have 6 molecules of 3-phosphoglycerate. This is our product for our first step in this pathway. Now let's check the source of each of these molecules here. For CO2, we know the source. I've explained that one earlier. Now for the compound that reacts with CO2, which is ribulose 1,5 bisphosphate. It is always in the plant. It is present in the cytosol of the plant. So they are making use of it from their archives. This enzyme that now catalyzes this reaction, which is ribulose 1,5-bisphosphate carboxylase, otherwise known as rubisco, that means the most abundant enzyme on earth. They are produced by the plant. Now, apart from this reaction that involves CO2, this particular enzyme can also bind to oxygen, which will lead to photorespiration. But in that process, no energy is being gained. So since there is no energy, it's just like a waste of process. Now, the products we get from this step 1, which is 6 molecule of 3-phosphoglycerate now, is now what we will now use in step 2 of this particular pathway. And step 2 is the reduction step of the pathway. So let's go to step 2. 6 molecules of 3-phosphoglycerate now will now react with 6 molecules of ATP to give us 6 molecules of 1,3-diphosphoglyserates. And the enzyme that will catalyze this particular reaction is phosphoglycerate kinase. So the next thing is that this particular product now is now also, we start our next reaction now. So 6 molecules of that product will now react with 6 molecules of NADPH. The enzyme that will catalyze this reaction is glyceraldehyde 3-phosphate dehydrogenase. The product we'll get from this reaction now is 6 molecules of glyceraldehyde 3-phosphate. Now, I want us to do like a short assessment of ourselves by answering this question. Now, this ATP and NADPH that was used in this reduction process of the Calvin-Benson cycle, where did we get them from? I'm waiting. I want you to answer before I give an answer here. So I trust you've gotten the answer right. That's good. Well done. So for those of us that are forgotten, these are the products that was gotten from light phase reaction in photosynthesis. The ATP was gotten through electron transport chain and oxidative phosphorylation, while the NADPH was gotten from photolysis through electron regeneration. If all this is sounding strange to you, please check the description. I've attached some of our previous classes that can be of help to you in understanding this process very well. So check it out so that you can understand this with us. Now, these are products here is what we are starting our next step with. The third step now, which is the isomerization step or stage now. You remember this? That's the product that we got from our step 2 or the stage 2, which is the reduction stage. We have, for these 6 molecules that we have, one of these molecules we leave the circle. One of those molecules we leave the Calvin-Benson circle, leaving us with 5 molecules of the aldehyde 3-phosphates. Which means that we are starting our third step, which is the isomerization stage, with 5 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate now will now undergo several complex enzymatically regulated reaction, which will now finally give us 3 molecules of ribulose 5-phosphate. So our product for the third phase now, or the third stage, or the third step, which is the isomerization reaction, is 3 molecules of ribulose 5-phosphate. We are now moving to the fourth step, which is the phosphorylation step. And also remember that we will be starting that step with the product of our from this our isomerization step. The fourth step, which is the phosphorylation. You remember this? This was the product that was gotten from our isomerization stage. So, and we are starting this particular step with it. So let's write 3 molecules of ribulose 5 phosphate will now react with 3 molecules of ATP, and this reaction will now be catalyzed by the enzyme phosphoribulose kinase, which will now give us 3 molecules of ribulose 1,5 bisphosphate. So you can see how it's flowing in a circle. Like step 1 will give us the product, and that is the product that step 2 we make use of. So when this one also give us a product, the product will be make use of the product will be make use by step 3, and when we finish step 3, whatever we also get from step 3 will now is now what we will use in step 4. And now our product for step 4 now will now come back to step 1. That is how this circle will now continue. So that's just everything about Calvin Benson's cycle. But there are some keynotes from this circle. First, the second step of this pathway, which is the reduction step, we were able to get 6 molecules of glyceride 3 phosphate, and we said one of them we move out of the circle, leaving us with 5 molecules of glyceride 3 phosphate. Now, the 5 molecules is now what we start our third step. Now, the question is, that one molecule that left the circle, what is the fate of that particular molecule? This one molecule that left from this circle moves into the cytosol, and from the cytosol it now enters into the gluconeogenesis pathway. If you've not forgotten all your pathway, now you remember that gluconeogenesis is a pathway that helps you generate glucose from non-glucose sources. So if this glyceride 3 phosphate enters, leave this carboxyl cycle, enters into the cytosol, and from the cytosol enters into this pathway, you know that what this pathway will give us is what? Glucose. You are correct, which is what we are looking for, because photosynthesis we make use of water and carbon dioxide to give us organic compound, and our organic compound is our glucose. So this is how we achieve our glucose. Now, this of glucose now, through all this process, the glucose will now be stored in the plant. So when the plant now need it, they will now break down the glucose. And what pathway will involve the breakdown of glucose to give you energy? You are right, glycolysis. So this glycolysis pathway, where they will now get energy, and they will now use their energy for several functions like cell growth, cell repair, resistance to pathogens, etc. We've mentioned all those functions in our previous classes on photosynthesis. So this is just the summary of the dark phase of photosynthesis, also known as the Calvin-Benson cycle. No, one complete turn of Calvin-Benson cycle now will first make use of how many carbon dioxide? 3 molecules of carbon dioxide. So make use of 3 molecules of CO2. Then it will now give us 1 molecule of glyceride 3 phosphate, which escape the cycle to go and synthesize glucose. Also, during this particular cycle, we make use of ATP. At step 2, we make use of 6 ATP, and also at step 4, we make use of 3 molecules of ATP. So in total, for this cycle, we make use of 9 molecules of ATP. So also in this cycle, at step 2, the reduction step, we make use of 6 molecules of NADPH. So for one turn of this, for one complete turn, these are what was used, and we're given this to eventually give us glucose. So now, if you are asked what is now the importance of this particular pathway, first, it helps in carbon fixation. So with that, it helps to control atmospheric carbon. You know, it is CO2 that we are breathing out. So if these plants are not fixing this carbon for us, we are in danger. So it is very important in carbon fixation. Then also, it is also very important for the plant to be able to synthesize glucoses, which will sustain their metabolism. As I promised earlier, I will now list out the 4 steps, then the products we are getting from each of these steps, and the enzyme that catalyzes the reaction involved in these steps. The first step is carbon fixation or carboxylation. Carboxylation. Then the enzyme that catalyzes the reaction that occurs here is ribulose 1,5 bisphosphate carboxylase, also known as Rubisco. Now, the product that will be gotten from this step, 6 molecules of 3-phosphoglycerate. Second step is the reduction step. The enzyme that catalyzes the reaction that occurs here are one, in the first reaction that involve this our product and ATP. That first reaction, the enzyme that catalyzes the reaction is phosphoglycerate kinase. Then the product that we will get from that first step of reduction is 1,3-diphosphoglycerate. 1,3-diphosphoglycerate, but we are getting 6 of that, so that's why I wrote 6-DPGA. Now, to the second step in that same reduction phase. Now, the enzyme that catalyzes the reaction between this our product and 6 molecules of NADPH is glyceride 3-phosphate dehydrogenase. This PS phosphate, I know you know that. So glyceride 3-phosphate dehydrogenase, and the product we are getting from there is 6 molecules of glyceride 3-phosphate. 6 molecules of glyceride 3-phosphate. And remember that out of 6 molecules gotten from here, one of them will leave the circle to enter into the cytosol, where it will enter into gluconeogenesis to give us glucose, while the remaining 5 will be used up in the third step, which is the isomerization reaction, to help in regenerating the cycle. So in isomerization reaction, we did not have specific enzyme that catalyze this reaction, but all we know that is a series of complex enzymatic reaction, but these enzyme reaction are regulated, actually give us 3 molecules of ribulose 5-phosphate. Now, for the last step, which is the phosphorylation step, the enzyme that will catalyze that reaction is phosphorybolose kinase, and the product we will finally get from this step is 3 molecules of ribulose 1,5 bisphosphate, which is what we started with in the first place. And with this, the cycle will continue. With this, we've come to the end of today's class, where we've extensively studied the dark phase of photosynthesis, focusing on the Calvin-Benson cycle process of glucose generation. See you in the next class. Bye bye.